All right, so we talked about HardyWeinberg equilibrium. It was a mathematical formula, if you recall, and it was used to calculate allele frequencies. Part of the point of the formula is not just that we can calculate the current allele frequencies, but if a population, if you read in a question that a population is in HardyWeinberg equilibrium, what it means is that that allele is stable, that it is not changing, that the frequency of organisms carrying that particular allele has not changed or is not in the process of going through a change. So in order for that to be the case, there can't be certain things going on in the population. So there are 5 things, and there's actually 2 ways you might see this on a test. You might see this as a question where they say which of these is required to maintain HardyWeinberg equilibrium, or you could see the question which of these would disrupt HardyWeinberg equilibrium, sort of opposites. So the way I've got them listed here is how they would disrupt HardyWeinberg equilibrium. So the first thing that would mess up HardyWeinberg equilibrium is mutations. In other words, in order to maintain it, you would need no mutations. If you have a certain frequency of, I don't know, a certain percentage of the rabbits are white and a certain percentage are black, if there's a mutation that causes more of them in the next generation to be white, or there's a mutation that causes a new color to appear, that would disrupt HardyWeinberg equilibrium. So you would need, in order to maintain it, we would expect there are no mutations in that particular gene. Secondly, no gene flow or migration. And by migration, this could be into the population or out of the population. So again, in order to maintain, you would want no migration. Now you might say, what do you even mean by, how does that affect it? Well, think about it this way. For example, gorillas live in groups. Usually there's one dominant male gorilla who's sort of in charge, and that male mates with all the females. Any other males that are born are always going to be subservient to him until at some point they mature and they leave and they form their own group. That's migration. So if some of the males that are carrying the gene for, large noses leave, now the frequency of large noses has changed, and there'll be less of that gene to be passed on to the next generation. A little note, immigration would not necessarily affect a population if the organisms that came in were beyond reproductive age, which is something they could ask you on a test. Which of these would, even if you do have migration, which of these would be least likely to affect the next generation? If the immigrants that come in are all 80 years old and they're not planning to have any children, they're not going to affect the next generation. Even though they may bring in new genes, they're not passing them on. So to keep HardyWeinberg, you want no mutations, you want no migration in or out of the population. The third thing that can disrupt HardyWeinberg equilibrium is non-random mating. In other words, in order for the population to stay the same for that trait, we have to assume that organisms are not specifically choosing mates that have that trait or don't have that trait. Here's an example of this. These birds, I know it's a little picture, they're called widow birds. They have really long tails. Scientists went out and what they discovered was females picked males with the longest tails. Those males got to mate more than the ones that had shorter tails. So obviously that's going to screw up HardyWeinberg equilibrium because the long tail trait is going to become more common as the generations go on. Now, interesting thing, you might say, how would that benefit at all that they pick longer tails? Like, what benefit is having a longer tail? And it turns out what they discovered was males that had longer tails, it wasn't the actual presence of the tail. The tail itself doesn't help the male. But in order to have a long tail, it means they had to not have parasites, because parasites make feathers fall out. They had to probably have lived quite a long time and not been attacked by anything, because that would have ripped out tail feathers. And so, in essence, even though they didn't know it, by picking males with long tails, the females were choosing the healthiest males. And so a lot of times it's not as obvious as we think, why instinctually certain traits are favorable. They may not seem like they're a beneficial trait, but the long tail in reality was sort of a marker for healthier males. The next one, small population size will mess up HardyWeinberg equilibrium because something can happen called genetic drift. I'm actually going to talk about this more on the next slide, so I'm not going to get into it now. But again, in order for HardyWeinberg to be maintained, you generally need a pretty large population. In a small population, genes can get wiped out just by random chance. And again, I'm going to go into genetic drift on the next slide, so I'm not going to talk about it much right now. And the last one is natural selection. Obviously, if having the trait makes you better fit, you're going to pass that trait on more. If having the trait is not good, you're going to pass that trait on less. So these are the 5 things that would cause a population to evolve. These disrupt Hardy-Weinberg equilibrium. Again, if you're asked the opposite question, which of these is necessary to maintain Hardy-Weinberg equilibrium, you want the opposite of that. You want no mutations. You want no gene flow. No migration in or out. You want random mating, that the females and males are randomly all mating with each other. They're not necessarily picking the males that have or don't have that trait. You want a large population size, and you want no natural selection for or against that trait. Those would be the 5 things that would keep a population in Hardy-Weinberg equilibrium. If the population is not in Hardy-Weinberg equilibrium, what happens is what we call microevolution, which is really what Darwin saw. Darwin never proposed the Big Bang Theory or said that everything came from a single bacteria that was created. He didn't know anything about any of that stuff. All Darwin said was, I'm in the Galapagos Islands, and I can see these gradual changes, and this ancient tortoise isn't here anymore, but I can see that over time it changed and changed and changed, and it looks now like the tortoise of today. That's all Darwin saw, microevolution. He did not propose, endosymbiotic theory with the mitochondria and how prokaryotes became eukaryotes and how a fish became a rabbit or anything like that. Anyway, so that's microevolution. Genetic drift. So remember I just said one of the things that will mess up a Hardy-Weinberg equilibrium is if the population is small, genetic drift can happen. So what genetic drift is it's a random process that happens specifically in small populations where a rare allele can get wiped out. Think of it this way. If you had a population and there were only 10 people in the population, and only, let's just say, one of them had blue eyes, it's possible that that one person doesn't happen to reproduce, or they get struck by lightning, or whatever. Bottom line is, the gene is gone. Now, even if only 1% of a population of 1 million people has blue eyes, 1% of 1 million is still a really big number. So the chances that just randomly a trait's going to get wiped out is going to be very small. But if the population is small, those chances are much higher. And so what can happen is an allele can actually become what we call fixed in a population, meaning every single individual is homozygous for that. That everybody in the population is big B, big B. Or everybody in the population is little b, little b. That's called being fixed. And if you were to start, and I'm going to show you in a second a computer simulation of this, if you were to start with only, say, 20 individuals in the population, each one might drift randomly in a different direction. The key words to know that they're talking about genetic drift, just so you know, on a test, small population, always a small population. And the other one is it has to be random chance. It has to be, if they say that the gene got wiped out because it was a crappy gene, because, having that gene made you less likely to live, that's not genetic drift. If it says that the gene disappeared because of random chance, that person got struck by lightning, like I said, or whatever, that the gene didn't disappear because it was bad, the gene just disappeared because of just random chance. So let me show you really quick. I have an animation of this. So this is a simulation of flies, and only 25 flies start in the generation. And notice the computer simulation. After a very short time, everybody in that particular population is big A, big A. Here it is again. After just a few generations, every single individual in the population is little a, little a. Here's another random simulation. Now everybody's big A, big A. Here's another one. Lasted a little longer, but everybody's little a, little a. And all they did was they started with flies, and they... every time they gave birth to offspring, they randomly picked 25. Then from their offspring, randomly picked 25. And it doesn't take very many generations, and everybody becomes homozygous for the trait. Now notice if I repeat it. This is now with 500 flies. Now notice my simulation. Does it drift a little off the middle? Yeah. But does it get even close to everybody being the same? Absolutely not. And I can do the simulation a whole bunch of times. And you'll see that I pretty much get the same thing every time. There's another simulator this one, and I'm recording and you can't see my what I'm doing. But here I choose a population size, let's say of 100 individuals, and it literally randomly simulates over time. And you'll see generation. I put 1,000 in last period, and it was over 200, 300, and it just kept going and going and going. But if I change this, and change the population to have, let's say, instead of 100 individuals, just 10 individuals. So let's just do it with 10. Ah, I didn't do that. Oh, I see. I have to do it this way. Sorry. I guess I could have typed it in. That's all right. All right, so I'm going to go down to, this time, we'll do it with just, now I'm going up again. I'm wasting recording space. I'm sorry, people listening to the recording. Oh! Yeah, I know. I can't make it. There. 9 individuals. Let's see what happens. There, 2 generations. The allele was fixed after just 2 generations. Yeah, so if I do it, let's say 25 individuals. Let's see what happens. Again, it's random each time, so it could be longer than one expects. Almost 14 generations. So having a small number of individuals, it's very likely that a particular allele could get either wiped out or everybody ends up homozygous dominant or, homozygous or recessive for that particular allele. So that is genetic drift. This is sort of a picture of it. Again, the green ones happen to get stepped on. Suddenly the gene frequency of green dropped way off. Not because green was bad. He wasn't looking for the bugs and saw the green ones. He just happened to step on green ones. There are 2 other specific kinds of genetic drift. Again, this is still small populations. One is called the founder effect. The founder effect is where the frequency of an allele is more common in a particular population because one of the original founders had that trait. One of the examples you may find online of this is the Amish. Since the Amish are a closed group and they tend to marry within their own community, and rather than bringing in outsiders, one of the founders had polydactyly, which is extra fingers. Today, 1 in 8 people are heterozygous for that trait in the Amish community. In the general population, it's a much lower number. But because one of the founders had the trait's more common in that population. And the other one is, I should say, the bottleneck effect. The bottleneck effect is where a natural disaster happens and almost everybody dies, which means that those few survivors, pretty much it wipes out all the variation, because now everything's going to get repopulated with just those few survivors. Imagine if some kind of worldwide global epidemic happened and everybody was killed off except some small little mountainous area of Sweden. Obviously, people would look a lot different 1,000 years from now if the entire Earth got repopulated just from that one small group from Sweden. All the variations of our hair color and our skin color and our eye color and probably a lot of other traits we don't even think about would be gone because of this bottleneck event. So I'll show you a picture of these. Okay, so this is a picture of the bottleneck effect. Imagine you have all this variation represented by the different colored beads. Some kind of natural disaster happens. You end up with only these few survivors. Now notice how the variation is limited. Now, could variation come back? Yeah, if mutations happen, we could get new variations. But right after, this is all that's left to repopulate. I think they say almost all living cheetahs are genetically identical. They think some kind of a bottleneck event happened that wiped out a lot of the variation among the cheetahs. And so there's not a lot of genetic variation in them because the few individuals that remained are what have brought back the population. And this is the founder effect. You have all this variety of colors of bugs. Not all this variety, there's 2 colors. But on the island chain, there's only red ones because the founder that moved over to the island was red. They show this one too. That you started with on the mainland, all these different colored flowers. A bird brings seeds to a new island, and now all the ones on the island are orange because that's what the founder was. So that's the founder effect. All right, wrapping it up. Actually, let me just hit this because